Hey, Lauren. Hey, Lauren. I'm not sure if you're getting started already, but I got questions, girl. Please.
Fair warning, guys, it's a little bit loud in here. I'm in the cafeteria as of right now, and I expect it to get louder. Are you guys like okay with this noise level? Yeah, of course. If you want to type it, I could probably hear it better, or I could probably read it better. Yeah, sorry. I'm gonna mute myself just to make it a little bit less. Okay, cool. I don't want it to be too loud. So, what questions do you have? Yeah, okay. Let me go ahead and pull up that PowerPoint just real quick. Get to the right section, and then I'll share my screen. Here we go. Share my screen. So, let me also see if the chapter 6. Okay. So the process of replication for bacteria specifically is a little trickier. Well, it's not trickier. It's a little more simple. We've only really learned about eukaryotic division throughout high school. So, and like other classes as well. I mean, anatomy and physiology is all about what we are, right? Not bacteria. So, okay. So we understand the structure of the circular chromosome, right? Like that's good. Remember that it kind of makes these little. Remember that it's like a rope, right? A rope that's twisted around itself, and if you start to unroll one part of the rope, the other part is going to be very dense. It's going to supercoil, right? So we have certain enzymes to prevent us from having certain negative effects, right? So let's see. I'm trying to get to the page where it's like the best. Okay, stop. Let's see. Do you have those 3 slides? Yes, I do. Okay, yeah, that would have been a better idea to pull those up. I'm just going to share my screen, period. I don't care if you guys follow my mouse or know what classes I'm in. Let's see what classes I've taken. Yeah, I have to agree with that one. The videos, however, they're pretty good. The videos are pretty good. Like, a good one for the DNA would be right here. But this, I believe, is it? I hope it's it. Okay. You know what's funny? This picture was actually on our exam. So if you want to quiz yourself later and like test yourself on labeling that, that'd be a good thing. Where the heck did it go? I think it's
I don't know if you can hear me, but it's there you go. It's right there under the chapter 7.
okay.
Under chapter 7, the PDF.
Here it is. I'm looking right at it. I knew it was here somewhere. So let me. So helicase comes in, right? Helicase and gyrase are a nice little team. They come in and they start to unwind. The helicase starts to unwind, and then the gyrase prevents that supercoiling I was talking about, right? So the helicase is trying to do its job, and the gyrase is behind it, kind of just keeping things neutral, right? And so this is our start, right? Moving on to our next phase. So our primase is going to come in. Our primase adds RNA. Primase is RNA. It's going to add on to the leading and lagging strand. Now, the reason why one's leading and one's lagging is simply because these things can only work in a 5' to 3' direction. And I know it's a little confusing because you're like looking at the lagging strand and you're like, well, it says 5' to 3' right there. You have to turn around and think about what direction the strand itself would be going. So when one side's a 3', the other side's a 5'. Okay, it's a little bit tricky, but if you draw it out, I feel like it helps a lot more. So our primases and most of our things, they're going to move 5' to 3' always. And this is just going to add, this is basically telling the like ditzy DP1 where to go. Making cast. Yes. Yes. So replication always happens in a 5' to 3' direction. It doesn't really matter what it specifically is replicating it, whether it be primase, DNA polymerase, any of those, they have to work in a 5' to 3' direction. Yes. That's why you see that error, and 5' is the base, 3' is forward. Yeah. Okay. So our primase starts this, right? It's the primer. Yeah, no problem. So our primer starts, and our DP3, our ditzy DP3, is like looking around where to go while the helicase is unzipping. And then all of a sudden the primase starts and signals for this DP3 to come in. The DP3 picks up where it leaves off and continues, right? Continues the transcription, but this time it's going to be DNA. It's not, there's no longer uracil instead of thymine. It's going to be the regular bases in DNA. Okay. So that's going to come in after the primer. Now, we don't want there to be random segments of RNA, especially because we have a leading and lagging strand. We don't want random segments of RNA to be in there. So DP1 comes in and replaces that primase, whatever the primase did. So DP1 comes in and replaces it, but now we have these weird gaps, right? You can see in between where the primase was and where the two DPs are working, right? So we have this gap now. So ligase, the glue, comes in and seals those weird little pieces, right? They're going to put them together and it's going to look cohesive now. Okay. And I think then that's basically the whole process. But what you guys really need to be able to visualize is here. Let me pull this up. So so you were looking at this, right? You saw this is the direction of replication, right? That's the direction of replication, and this is your lagging. And this is your leading, right? So if we have a lagging and leading strand, and you saw how the primer was added here, and then this was the DNA, by the DP3 action here. So you saw how it actually had to go this way because this is the 5' here of the strand, and that's the 3', but it has to always go 5', 3' on the replication side. So this is the leading because if you look at it, right, this is 3' down here and this is 5'. So when it adds, just to give you a little bit of a clearer image, when it adds on, right, this is our base strand, it's going to just have that little piece and continue going forever, right? It doesn't, the DP3 doesn't need to stop. It just keeps going in the direction of the replication. Now, what happens to the lagging strand is if we open it up a little bit more, right? The helicase opened it up. This is where we started, right, with that primer, and then this is what the DP3 did. So this is what the primer did, but it opened up even more, right? For this one, it's just going. For this is the leading strand, this is the lagging strand. What has to happen now is another primer has to come in, prime another section, and then DP3 has to jump back down here and work again. And then ligase, ligase, ligase. DP3 stops because of the lagging effect since it's going in the opposite direction of replication. Essentially, yes. So DP3 essentially doesn't have anywhere else to go. Remember, it's a ditzy thing. You know, it works for so long, runs out of work. It just kind of like, I don't know. It doesn't really, you know, say, oh, well, they need my help over there. Now I need to go replicate down there. It needs another primer to come in and tell it where to go. So what winds up happening is, you know, how we were talking about the whole process with, you know, first comes helicase and gyrase helps. Helicase unwinds, and then the primer comes in. The primer comes in, adds RNA, then all of a sudden the DP3 comes in. And then the DP3 continues where the primer left off. The DP1 comes in, replaces the primer, and then ligase glues it all together, and now we have a completed strand. That has to happen over and over and over and over again for this lagging strand. Where the primer comes in? Yeah. So let me go back up here because hers is a little thinner. So, right, we started here where our helicase comes in and unwinds. Our DNA knows when it's being unwound and when it's time for all this to happen. Remember, they're always communicating. Gyrase is just kind of chilling, doing its job on its own, doesn't really need any help. Helicase unwinds. Now primase knows we're unwinding. It says, okay, now I need to prime it. This is my time to shine. So it's going to come in and it's going to either start at the end for the leading strand or start at the replication fork for the lagging strand. So then they're going to work 5' to 3' as per usual. And that primase is just RNA that tells the DP3 like, hey, come on over here. Remember, DP3 is ditzy. It doesn't know what's going on. Where do they start at? So where does the primer start? Okay, so the primer, it just kind of depends. The primer starts for the leading strand just at the start of the replication. So whatever is unwound already. So if here, I'll clear this off. Maybe. And that's kind of, can I like erase it like this? And no? Okay. Well, I'll just kind of try to describe it. Let's see. So think of it like a bubble, right? So this bubble that's happening, here, I'll pull this up actually. You see kind of here where the helicase has done its job and unwound, but this is still wound on this side. So what happens is the primer, the primase for the leading strand will start here and head in the correct direction and it'll keep up as helicase keeps unwinding. So it'll just kind of follow, right? And it'll be a little bit faster than the lagging strand. For the lagging strand, it has to go in the opposite direction. So if this is the direction of replication, we have to focus here and say, okay, the primer is going to start here, and then DP3 is going to come in. Now this section's unwound. So now it's got to prime down here, and DP3 is going to come in. And it all just kind of works simultaneously. So the primer, it kind of just depends at what point we are in replication, but the primer will start at the greatest, like the most opportune time. Does that make sense? Okay. So yeah. Sometimes in order to like memorize this stuff, you're good. Sometimes to memorize this stuff, I give them personalities. It makes me think about how they interact with each other, and it makes me remember it. So if I say that DP3 is ditzy and it doesn't know what to do, and it needs primase to tell it where to go, then I know that primase is going to trigger the reaction with DP3. So if you think of, you know, like the person who's chronically late in your friend group, like that would probably be DP1, because it kind of comes in like late. It's like, oh, I was supposed to be here and I was supposed to code this DNA. And then, you know, those people who are like the mediators in every fight, and they like prevent the friend group from like breaking up all the time. You can say that's ligase, that's the glue, right? Like just doing things like that, even if you want to do different relationships, that's fine. But things like that will really help you. And I also recommend drawing each of these out. These are really good drawings. And if you want to do it on like a piece of printer paper and make it your own colors, make it fancy, make it look how you like, you can. I mean, this is for you guys to visualize it and for you guys to see the steps. So how are we feeling? Now, this is a really important part of the exam. What do you think would be best to focus on for the quiz? I feel like I've been cramming as if it were an exam. Okay, so I know for a fact that chapter 6 and like most of chapter 7 is going to be on the quiz. So I was told that you should know about, okay, let me pull up the PowerPoint. So I was told you should know about microbial growth. You should know about logarithmic representation, and this. Know this. There will be at least one question about it on the quiz. So if you know this and how each of these functions in a like colony, then you're good. Did you want me to go over this? Okay. Yeah, no problem. So think of this in terms of your colonies, right? You go to your plate and you have a colony. So you have your lag phase, right? You have bacteria growing, and they're growing and working on themselves. They grow and work on themselves, and all of a sudden they are like, man, I want to reproduce. So then we have this exponential growth, right? It's this idea of the generation numbers, right? You just double, 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 double. All of a sudden, you're at 1 million and you don't even know it. Also, you're good, Amanda. Have a good day. But anyways, so we have this exponential growth where they're just reproducing, reproducing, reproducing. All of a sudden, you know, there could be multiple reasons why this happens. It could be, you know, programmed cell death. It could be nutrients are running out. It could be space. It could just be competition, right? But all of a sudden, the growth stops. And that's where we have the stationary phase. Once all the, either all the resources have been used up, it just depends. Once that happens, we're going to see the death. We're going to see the death of the whole thing. And eventually, at the very end, there won't be any bacteria left because they've all died. There might be a few, the select few that are genetically hardy and strong, but usually they just die. So I think the most that you'll need to know about at least this is the lag phase is first, the log exponential growth is second, then we have the stationary phase and the logarithmic death. If you were asked about a question that would require you to critically think about this, I believe you would be able to. But if you were to be asked a question about this and she does not give you a graph already, draw it out if you can. And it'll help you on the quiz. But yeah. So are you okay on that one? Okay, good. Cool. All right. So it's going to be a little bit more, it's going to be all of chapter 6. Just know that. You'll probably want to know this, at least in a broader schematic. Let's see. You'll want to know the mesophiles and the psychotrophs. You want to know what those specifically do. So the mesophiles are disease-causing, psychotrophs are food spoilers. And just understand what this graph is telling you, like the information it can give. But I believe that you could figure it out right there. Let's see. Let's see. Thermophiles, obviously they like heat. You'll obviously by the name you know what a acetophile is. Let's see. I think all you really need to know also is that Helicobacter pylori, is the one found in your gut. That's an acetophile. Let's see. Cause ulcers. Alkaphiles. Let's see. Oh, in terms of this, know your osmosis and your solutions. So remember that in any kind of solution, when it says hypotonic, that means that there's a solute in the water, and if it's hypotonic, that means that the solution has too little solutes. So the solutes are coming in, or I'm sorry, the water is coming in. The water is coming in to try to make the solution around more equal. So if you want to make something equal, right? If we have too much solute and not enough water, in order to make it equal, we either need to get rid of some of the solute, which can't happen, or get more water. That's what our hypertonic solution is, right? So if we want to get more water, we have to pull that out of the cell. And if we pull it out of the cell, what happens? It triples, right? So in a hypotonic situation, we have too little solute and too much water. So all of a sudden, in order to make this a balance, we have to bring the water level in the solution down, which means that we have to flood the cell with water. I think that's one of the things that tripped me up a lot and tripped other students up a lot as well. Let's see. Do you know what a saprophyte is? I hope you do, because that's going to be on the quiz. Sacrifice. Here, I'll type it out. I believe that's how you spell it. Don't quote me, but it's okay. It's just an organism that has to consume other organisms for food. So but the thing is, it only consumes dead organisms. I'm trying to find the slide that would say this. I can't. So yes. So like you just needed to trigger the memory, but just they eat dead things. That's basically it. And you might be asked a critical thinking question in terms of like building off of that idea. I believe so. If I were to ask you a question like, what is a thermophile's optimum growth temperature? And I said 0 degrees Celsius, 37 degrees Celsius, and 97 degrees Celsius, what would you say? There you go. That's, I mean, like questions like that will be basically for the optimal growth temperature, the growth chart, and then I think the only other thing you'll need to know is, and there's a lot going on. All you'll really need to know otherwise is the mutations. Let me find the mutations. Those are really easy to ask questions about. Yes. So let me, don't look at the screen, don't look at the screen, I'm going to try to quiz you. Okay, so tell me, or you can type it out, what a silent point mutation would be.
Can you hear me?
Yes.
Okay, that's even better, just because I'm walking out of the house and I can't really type. A silent is a point mutation is something. Point mutation is that it means it's a change in a single nucleotide, but it doesn't show any difference to the phenotype.
Yes, very good. And that's usually because of the redundancy in this chart that you'll see.
Right.
So the redundancy here is basically why silent mutations happen. Okay. And then what about a missense point mutation?
Missense means it changes it, a single nucleotide, and it changes it to like a stop codon. So it makes it like trunculate, like a shorter than usual DNA strand.
For the missense, you're close. The missense is
right. It changes the nucleotide or the amino acid. Did not make sense. It changes the amino acid.
Yes, it just changes it to something else, and it usually results in a non-functional protein. But what you were thinking of was a nonsense point mutation. That is when, you know, this ribosome is about to start the race with trying to make an amino acid chain, and then all of a sudden somebody comes in and says stop. It's like, what? No, I'm supposed to go. This makes no sense, right? So that's how I remember that. What about the frame shift point mutations?
Frame shift is a nucleotide is either inserted or deleted, and then that shifts everything over either to the right one, so it all changes all the amino acids, or to the left one, still changing the amino acid.
So one thing to remember about point mutations is it's always going to be about one. When you say it's an insertion or a deletion, you're right that frame shift can be either insertion or deletion, But if you say, you know, if you're going to look at an exam question and say, and it's going to ask you what type of genetic mutation this is, and you say it's insertion or deletion, it could be you know, it could not make perfect sense because that's in reference to more than one base pair. But if you say it's a frame shift point mutation, specifically in insertion or deletion, then that would be the most correct answer. But yeah, this is all you'll need to be responsible for, these 4/5 for the genetic mutation. All right, and I think that's all I have. So do you have any other questions? Okay. Okay, and is this your first PAL session with me? No? Yeah. Well, then I'll find your, I just need your student ID number. I'll find it though, because I write them all down. But I'm glad I could tell, and ooh, yeah. Thank you. Okay, I'm gonna write that down really quickly, but you are free to go. I'll be in class today, okay? Thank you. Have a good one.